every protein begins as something surprisingly simple, a chain. Inside a living cell, a ribosome reads genetic instructions and links amino acids together in a specific order. At that moment, the new protein is not yet the elegant molecular machine biology needs. It is a flexible, unstable strand, more like a loose thread than a finished tool. Protein folding is the physical process that changes that chain into a three-dimensional structure. That shape is what allows the protein to become active. Without folding, the chain may contain the right ingredients, but it does not yet have the right form. In biology, form is function. A protein works because its shape lets it touch, hold, move, recognize, or transform other molecules in exactly the right way. The folding instructions are hidden in the amino acid sequence itself. This sequence is called the protein's primary structure, and it determines much of what the final shape can become. Each amino acid has different chemical properties. Some prefer water. Some avoid water and tend to bury themselves inside the protein. Some carry electrical charges. Some are bulky, flexible, rigid, or chemically reactive. As the chain forms, these amino acids begin interacting. They attract, repel, twist, and pack against one another. A protein does not fold randomly into any possible shape. Its sequence creates a landscape of possibilities, making some arrangements stable and others unstable. The native state is the folded arrangement that gives the protein its biological role. Folding often starts before the protein is even complete. As the ribosome builds the chain, the first amino acids emerge while later parts are still being added. Those early sections may already begin forming small structures, creating contacts, and limiting what later parts of the chain can do. This means protein folding is not always a separate step after construction. Construction and folding can happen together. That timing matters because early choices can guide the rest of the process. A section that folds correctly may help the entire protein find its final structure. A section that gets trapped in the wrong arrangement may create problems later. Inside the cell, folding happens in a crowded environment where timing, chemistry, and local conditions all matter. When a protein reaches its native state, it has a defined three-dimensional structure, but that does not mean it becomes frozen. Many proteins need motion to function. Some regions remain flexible or even partly unfolded while the protein is still biologically useful. This flexibility can allow a protein to bind another molecule, change shape after receiving a signal, open a channel, or release a chemical product. A useful protein is not always a rigid object. It is often more like a tiny machine with moving parts. The folded structure gives the protein stability and organization, while its dynamic regions allow controlled movement. Life depends on both, enough order to perform a precise task, and enough flexibility to respond to changing conditions. The speed of folding varies dramatically from one protein to another. Some small proteins, especially single-domain proteins with fewer than about 100 amino acids, can fold very quickly. For them, milliseconds are common, and the fastest folding reactions can finish in just microseconds. These proteins usually have simpler structures, so the important contacts can form without a long search. Larger or more complicated proteins may take much longer. Some need minutes or even hours when studied outside the cell. They may pass through several intermediate states, almost like checkpoints on the way to the final fold. Folding time depends on size, contact order, and the complexity of the chain's topology. The path matters as much as the destination. One reason some proteins fold slowly is proline isomerization. Proline is an unusual amino acid because its structure can force parts of the chain into particular angles. Certain bonds involving proline can switch between different orientations, but that switch can be slow. If the protein needs the correct orientation before it can finish folding, proline becomes a bottleneck. This helps explain why folding is not simply a matter of chain length. Two proteins might have similar sizes but very different folding times because their sequences create different obstacles. The amino acid order defines the final structure, but it also defines the route. Some routes are direct and fast. Others require pauses, rearrangements, and waiting for specific chemical changes to occur. A correctly folded protein can do things that a loose chain never could. Folding creates surfaces, pockets, grooves, and charged regions that let the protein interact with specific partners. An enzyme, for example, may have an active site shaped to hold certain molecules in just the right position for a reaction. A signaling protein may have a surface that changes when another molecule binds to it. A structural protein may rely on repeated patterns that make it strong. In each case, the sequence becomes useful only after it becomes shape. The folded protein turns chemical information into physical action. This is why protein folding is one of the central links between genes and living function. But folding can fail. When a protein does not reach the correct native structure, it is often inactive. It may be unable to bind its partner, catalyze a reaction, or maintain stability. Sometimes the problem is worse than simple inactivity. A misfolded protein may expose sticky regions that should have been buried inside. Those exposed regions can attach to other proteins, forming clusters or aggregates. The new structure may interfere with cellular processes or become toxic. Cells have quality control systems to refold or remove damaged proteins, but those systems are not perfect. If misfolded proteins build up faster than the cell can manage them, they can become a serious threat to health. One of the most important consequences of misfolding is the formation of amyloid fibrils. These are long, ordered fibers made from misfolded proteins that stack together in repeating patterns. Amyloid accumulation is associated with several neurodegenerative and other diseases. The danger is not only that the original protein has lost its normal job. The new aggregated form can damage cells and tissues in its own way. Some misfolded proteins can even behave like infectious agents. These are called prion. A prion is disturbing because its abnormal shape can encourage other copies of the same protein to adopt that abnormal shape too. The genetic sequence may stay the same, but the structure spreads like a corrupt instruction. Protein structure also matters for the immune system. Antibodies often recognize three-dimensional features on a protein's surface, not just the list of amino acids inside it. If a protein folds differently, the surface it presents can change. That can affect whether the immune system notices it, ignores it, or reacts inappropriately. Some allergies are connected to this problem of recognition. The immune system may not produce antibodies for certain protein structures, or it may respond to a shape in a harmful way. This shows how deeply folding influences biology. The same chain can create very different molecular signals depending on its final form. Shape determines what other systems can see. Proteins can also unfold after they have folded. Denaturation is the transition from a folded state to an unfolded one. It can happen because of heat, chemical stress, burns, cooking, or disease processes. When an egg white turns solid and opaque during cooking, protein structures are being disrupted and reorganized. The interactions that held the original fold together are no longer stable. Sometimes denaturation is reversible, but often it is not, especially if unfolded proteins stick together and form aggregates. Even an unfolded protein may still contain residual structure. These leftover patterns can sometimes act as starting points for refolding, guiding later steps. Unfolding, like folding, is a structured, physical process rather than pure chaos. Protein folding has challenged scientists for decades because it sits at the intersection of biology, chemistry, physics, and computation. Since the late 1960s, researchers have tried to understand how a sequence can reliably find a useful three-dimensional structure. The challenge is enormous. A chain could theoretically explore a vast number of possible shapes, yet many proteins fold quickly and consistently. That means the sequence strongly guides the search through the folding landscape. Simulating this process is difficult because every atom, bond, charge, and interaction can matter. But solving it has huge value. If we can better predict and control folding, we can understand disease, design new proteins, and see more clearly how life turns molecular information into action. If you found this helpful, please like this video and subscribe for more explainers.